Imagine, for a moment, humanity not just, visiting space, but establishing genuine, thriving communities far beyond Earth. It's arguably the grandest challenge we face, transitioning from these brief, sort of fragile visits to building permanent homes out there in the cosmos. But how do we make space not just habitable, but actually truly alive? That's our deep dive for today. We're going to uncover the surprisingly central, yet maybe often overlooked, role of tiny organisms. Our sources call them microbial farms, and they seem key in constructing these future space habitats. We'll be exploring some fascinating insights from expert sources about, engineering life specifically for off-world environments. Our mission really is to understand how specific microbes can act as our biological allies, how they can transform harsh extraterrestrial conditions into self-sustaining ecosystems. This biological partnership is, I think, what will enable a lasting human presence in space. It's fundamental. So let's unpack this. Let's introduce our first microbial champion, Deinococcus radiodurans. It's often dubbed the radiation-proof biotech workhorse, and, for I imagine, very compelling reasons.
Oh, indeed. What's so remarkable about Deinococcus radiodurans is its unparalleled resilience. It actually holds the record. It's the world's most radiation-resistant organism known.
Wow. The most resistant.
Yeah, think about that. It can withstand cosmic radiation levels that would be absolutely lethal to most life forms we know. It can survive the harsh vacuum of space and endure extreme dehydration. This makes it, essentially, an ideal biological chassis. A really robust living platform for any bioengineering we want to do in space. Precisely where most other life simply couldn't make it.
Okay, it sounds like an organism almost designed for the cosmos. But surviving is one thing. What can this incredibly tough microbe actually do for us up there? How does it actively, contribute to building a habitat?
Right, good question. It has several critical applications, moving far beyond just, hanging on. First, it's a candidate for bioremediation of space waste.
Okay, bioremediation.
Yeah, engineered strains of D-radiodurans, they can metabolize toxic materials, things like heavy metals or perchlorates.
Perchlorates?
Yeah.
Right? Those are all over Mars, aren't they?
Exactly. Those salty, often quite corrosive compounds found abundantly in Martian soil. So imagine, a microscopic cleanup crew processing hazardous waste right there.
Okay, that makes sense. What else?
Second, it's promising for radiation shielding production.
Growing shielding? How?
Well, we could engineer it to produce radiation-absorbing biofilms or perhaps pigments like melanin or cytonamin. So it's effectively growing our own biological protection against those cosmic rays.
That's incredible. And you mentioned a third thing.
Yes, and perhaps most powerfully, it functions as a biofoundry. Basically, a genetic platform uniquely suited for these extreme environments. This means it's like a living factory. We can insert different genetic blueprints, allowing it to churn out specialized proteins or even perform gene editing, all within those incredibly harsh space conditions that would destroy most other organisms we might try to use.
It's astonishing how much utility can be packed into a single organism, especially one so resilient. Really makes you rethink what's possible for, on-site manufacturing. Okay, from that super tough radiation specialist, let's now pivot to something described as incredibly versatile. Bacillus subtilis. This one is called the robust multipurpose producer.
That's a very apt description. Its unique advantages for space exploration, they primarily stem from its ability to form spores.
Spores, right, like suspended animation.
Sort of, yeah. These spores can survive vacuum, intense UV radiation, and desiccation for extremely long periods. Years, potentially. This means we can store it, transport it, and then deploy it easily. Basically bring it to life whenever and wherever it's needed. Plus, it's remarkably easy to genetically modify, and crucially, it grows on a wide variety of substrates.
That seems key, given how limited resources will be.
Absolutely. That's critical when you can't just order more supplies easily.
The range of applications for Bacillus here genuinely broadens the scope of what a microbial farm could achieve. It's not just about one function, is it?
No, not at all.
It seems poised to solve multiple problems at once.
Exactly. Its applications are incredibly diverse. For astronaut health, for instance, it can be engineered for nutrient and probiotic production. Think synthesizing essential vitamins right there in the habitat like B12, K2, amino acids, or even antimicrobial compounds.
Okay, so direct health support. What about materials?
Yes. Then there's bioplastic and material synthesis. Engineered strains can produce biodegradable plastics like PHA.
PHA.
It's a type of polymer. It essentially allows us to grow our own space-friendly plastics instead of shipping bulky materials from Earth. It can also produce enzymes, biosurfactants for in situ manufacturing, again, significantly reducing our reliance on those costly resupply missions.
Just growing plastics. That's
Yeah.
still amazing to think about.
And finally, B. subtilis is a strong candidate for biomining.
Biomining, using microbes to get metals.
Precisely. Using biological processes like bioleaching to extract valuable metals from asteroids or maybe lunar regolith.
Wow, picture getting your raw materials from space
Yeah.
using just a microbe.
It opens up a lot of possibilities for ISRU in situ resource utilization.
So we have protection from radiation, the ability to produce materials and supplements. Now, for that vital task of keeping us fed and healthy in space, we turn to our Martian plant ally, Methylobacterium spp. What's its story?
This microbe has a fascinating connection to our current space endeavors already, actually. Methylobacterium spp, it turns out, is already found on the International Space Station.
Really? It's up there now?
Yep, particularly associated with plant growth chambers. This microbe thrives on methanol and formaldehyde. These are volatile organic compounds, VOCs basically, airborne chemicals that are pretty abundant in synthetic environments like enclosed habitats.
Ah, so like the kind of air you might find inside a spaceship or a base.
Exactly, and that makes it useful.
So if you've ever wondered how plants will grow efficiently on Mars or, in any closed-loop space habitat, this little organism holds a key solution.
It certainly does. Its primary role is as a plant microbiome booster. It enhances growth, stress resistance, and even nitrogen-fixing capacity in crops.
All critical for closed-loop life support, right? Growing food sustainably.
Absolutely critical for developing those sustainable closed-loop systems. But beyond that, it helps with air and surface detoxification. It breaks down those toxic volatile compounds we mentioned, effectively helping to purify the air in enclosed habitats.
So it helps plants and cleans the air.
That's right. And some strains can even aid in biowater filtration. They form biofilms that could potentially purify or condition recycled water. It's really about creating a truly circular, regenerative system that minimizes waste and maximizes resource use.
When you consider these incredible microbes individually, it's impressive. But let's zoom out a bit. What does this all mean when we put them together? What are the larger, system-wide benefits for space exploration and maybe colonization?
Well, if we connect this to the bigger picture, these microbes working together unlock some profound system-wide benefits. First and foremost is in situ resource utilization, or ISRU, as we touched on.
Using what's already there.
Exactly. They drastically reduce our dependency on Earth resupply. They do this by converting local substrates, things like regolith, the loose surface material on planets or moons,
Yeah.
atmospheric carbon dioxide, even harmful radiation itself into valuable resources. Food, materials, clean air, water. This is a fundamental shift towards true self-sufficiency in space.
Which is the ultimate goal, really.
Right, and this capability also enables autonomous biomanufacturing, the on-demand production of food, materials, maybe even drugs, biosensors, directly in orbit or on planetary surfaces, minimizing what needs to be shipped. What's also fascinating, I think, is that these microgravity farms act as synthetic biology test beds.
Test beds, how so?
Well, it's these extreme environments are perfect stressors. They push the boundaries for advancing programmable life systems for evolutionary research. We can learn things about bioengineering and life itself that we probably couldn't easily learn down here on Earth.
Using space as a lab for life engineering.
In a way, yes. And ultimately, these microbes are the pioneers for what you might call terraforming infrastructure. They help prepare alien environments, they can enrich soils, sequester harmful compounds, basically laying the biological groundwork for future, larger-scale colonization efforts.
When you combine these ideas, it paints a truly visionary picture for our future in space. Let's try and paint that picture for you, our listener. Imagine an autonomous biomodule. Maybe it's on a lunar base or a Martian outpost, perhaps even an orbital colony someday.
Yeah.
How would these three microbes, the ones we've discussed, work together in this integrated system?
Okay, yeah. In such a system, you'd see a dynamic, interconnected interplay. It wouldn't just be three separate functions. You'd have Deinococcus radiodurans hard at work, processing radiation-heavy waste, maybe recycling rare elements from the regolith using its resilience. Simultaneously, you'd have Bacillus subtilis tirelessly producing, say, structural bioplastics for habitat repair or expansion, alongside essential nutrition supplements for the human crew. And then Methylobacterium would be supporting thriving greenhouse crops, ensuring a fresh food supply, while also actively cleaning those VOCs from the air, maintaining a healthy, breathable environment inside.
So they're all interdependent in a way.
Exactly. This tripartite microbial farm isn't just about individual functions adding up. It's about converting fundamentally harsh space environments into living, self-regulating systems. These microbes are forming the earliest foundations of true space ecologies. It's quite profound, really.
So the core takeaway here seems to be that to genuinely establish a human presence beyond Earth, we need more than just, shipping supplies. Food, water, equipment. We need robust microbial allies that can actively co-create and maintain a livable biosphere with us.
You've hit on a key point there. By strategically farming Deinococcus, Bacillus, and Methylobacterium, and likely others down the line, humanity takes a critical and perhaps unexpected leap toward building self-sufficient space habitats, towards enabling sustainable colonization. These microscopic life forms are, quite literally, the living building blocks of our off-world future.
So a final thought to leave you with. What does a biologically empowered civilization beyond Earth truly look like? And how might these microscopic pioneers shape not just our future in space, but maybe even our very definition of life itself? Something to ponder.